Hi there, this is poster 583.09, and my name is Brodie Badcock Parks. I'm a master's student in the Department of Psychology and Neuroscience at Dalhousie University, and I'm really excited to get to spend the next few minutes to talk to you a little bit about the work that we've been doing studying doublecortin in songbirds. New neurons in the songbird brain express the endogenous microtubule-associated protein doublecortin, or DCX. And this protein is expressed in immature neurons only and is critical for neuronal migration. And doublecortin can be stained for using traditional immunohistochemical techniques to target new neurons. And this technique has been widely adopted to study neurogenesis in birds as well as in other vertebrates. But as you can see here with the images presented on the right, DCX positive cells have varying cellular morphologies. And as a result of this, the way that these cells are described or characterized varies in the literature. Now, the way that these morphologies generally have been described or characterized is by using categories. However, the number of cell categories or cell types described between studies varies, making consistency of findings and overall comparability quite difficult. So for the purposes of this project, we were specifically interested in whether 3 categories would be sufficient to classify DCX positive cells. And the 3 categories we chose were fusiform, multipolar, and round cells. And we came up with these 3 categories and how to define the cells that fit within each of these categories based on the previous literature. We defined fusiform cells as having an elongated cell body with 1 to 2 distinct straight or spindle-like processes. We called multipolar cells the cells that had a spherical cell body with multiple distinct processes and branching. And we defined round cells as having a spherical cell body with no or indistinct processes. So to test whether these 3 categories would actually work to classify DCX positive cells, we collected over 300 images of DCX positive cells from the songbird brain from multiple labs around the world, from multiple bird species, from different brain regions, and from both sexes. And we designed an online classification survey where participants were presented with each of these 300 plus images, or one of several negative control images, and they were asked to classify the cell based on the definitions that I just presented to you. And what we ultimately wanted to know was whether we could get over 90% of the cell images in our survey categorized with high agreement among all of our participants. And we defined high agreement as greater than 80%. We were also interested to see whether there were differences in categorization between participants who had previous experience working or not working with doublecortin. Ultimately, the results of our initial survey showed us that we could not meet this 90% standard. Only 87.5% of the images in our survey had high agreement among our observers, and that the disagreements in terms of cell classification here was not driven by the participants' previous experience working with doublecortin. And these findings ultimately suggested to us that our definitions, or maybe even our overall classification scheme, needed to be reworked. So what we opted to do was first to look at our definitions, and the way we did that was we stepped back, and we asked ourselves, which morphological characteristics can reliably distinguish DCX positive cells between our different categories? So what we did was we went back through all of the 300 plus images that we presented in our survey, and we scored them for each of the following morphological characteristics: cell body shape, number of processes, process branching, process direction, and process length relative to the cell body. And as you can see here in the panel on the right, we started to see differences. For example, if you look at the multipolar and round cells, you can see that multipolar and round cells generally have a circular cell body shape, and this is a trait that is generally not seen in fusiform cells. Likewise, fusiform cells have a rather elongated cell body, and this characteristic is generally not observed in multipolar or round cells. So using these scoring data, we then created a recursive partitioning decision tree with these morphological characteristics in mind. And what this is essentially is a workflow. So let's say you're presented with a DCX positive cell. This decision tree tells us to first look at the number of processes. If the cell has greater than 2 processes, then this is, in all likelihood, a multipolar cell. If the cell has between 0 and 2 processes, we then need to look at process length going down the decision tree. If the processes are shorter than the overall cell body, our cell is then in all likelihood a round cell. Now, if its processes are greater than or equal to the cell body in length, then we need to go down the decision tree and so on and so on. Now, what was really interesting was that now 94% of the images from our initial survey could now be correctly categorized using the specific decision tree model. And when you compare this 94% to the 87.5% finding from our initial survey using our original definitions, this suggested to us that in order to get to over that 90% benchmark of our images being correctly categorized with high agreement, these specific morphological characteristics and the order in which these characteristics are presented needs to be considered in our definitions. All right, so in terms of next steps, we've already rewritten our definitions, which you can see here on the right, using the scoring data, the decision tree data, as well as a few other findings from random forest model analyses. We've re-administered the survey, or we're in the process of re-administering the survey to previous participants, as well as new participants, in the hopes that these updated definitions can increase the number of cells categorized with high agreement from 87.5%, which was the finding from our initial survey, to over 90%. And remember, the overall objective here is to create a new classification scheme that can be adopted, ultimately increasing replicability across the literature. All right, so wrapping up, big thanks to all of my collaborators at Dalhousie as well as at McGill for their work on this project, especially to Connor Stadnyk, who led and coordinated this work as part of his undergraduate honors thesis. Big thanks also to Dr. Jacques Balthazar, Dr. Tom Smulders, Dr. Scott McDougall-Shackleton, and their teams for contributing images of DCX positive cells that we used in our surveys. If you have any questions for me, my virtual session is on November 10th from 3:30 to 4:30 Central Time. And if you're actually interested in completing the next iteration of this survey, please get in touch via email or via social media. I'd be happy to send you the link. Thanks very much for your attention.